Okay, good morning.
Good morning.
Okay, let's start our lesson with a revision about the previous lesson about moving genes. Can you give me a summary about moving genes? How can
The genes,
genes
like,
move from?
when the genetic engineers, they move the genes, and it's hard to do it, so like, they need skills to do that.
Okay.
And it's easier to use bacteria because they don't have a liquid. Like, their genes are just in a string it's loose inside.
Okay, what about the natural process? Before dividing any cell, the nucleus needs to copy itself, which is copy the genes. So in this way, genetic engineers use this method, dependent on this method, to use it in copying other genes or any genes that they want in other organisms to make it easy to extract it later. So living things can be genetically engineered to make useful products by adding genes from other organisms to them. Good morning. Such as the artificial spiders or the spider silk. They were using the spider silk's genes makers and they were injected them or adding them to some kind of bacteria, which is the salmonella, and they make it to copy it or make it to reproduce, and then they will throw away or throw out the silk. And also in farming, they were using this method in cows and goats to produce the genes that they want. But this takes too much. But the fastest way, which is adding to the bacteria. Today we will learn about, or we will study about using genes. How can scientists use genes, or how they used genes in science to save some lives, to change something, to produce something. Okay, so we will start about malaria disease. Okay, we will study today about how can we use genes to cure some diseases that have resistance against medicines. When we talked about, or when we explained about the resistance, do you remember what is resistance?
It is like the mice, when they got used to the medicine, they're not affected from it any longer.
So when the body or the organism gets used on something, so it will fight it. It will be strong. It will develop features against this disease or against medicine or against any danger, any possibility of danger. So this called resistance. Okay. Also the malaria developed a resistance against the medicine. Okay. So the malaria disease is getting stronger, but scientists try to stop it. So can you give me can you give me an example of, a summary about the malaria disease, about the symptoms, about the causes? What causes the malaria disease?
It comes from the mosquitoes.
Okay.
Special type of mosquitoes.
Okay. And how can this disease transfer by this mosquito?
Blood, transferring blood.
Okay, what this blood has?
Malaria.
No, the malaria is a disease, okay, but it has a protozoa that cause the malaria. Protozoa, which is the simplest organism, which is a parasite. It's not a bacteria or a virus. It's a parasite that feed on the blood, okay? So this protozoa comes in the mosquito. Mosquito has it, so it will transfer it when it's biting or injected during sucking the blood from one person to a person, okay? Do you remember the symptoms?
Fever.
Okay, fever.
Fever. Okay.
When someone has a fever, high fever, what do they feel?
They can't walk. Lazy.
They feel
Okay.
sick.
No, they have something in their body. They have
Low
pain.
blood pressure.
Pain. They have muscle pain. If you get high temperature, you will feel muscle pain, right? You are paining, okay? They will feel muscle pain and chills, which is like shaking, feeling cold, okay? These are the symptoms of the malaria. Okay, here, the protozoa of the malaria became resistant against the diseases because it's getting stronger. So it's getting resistance to this disease. But when they survive, they reproduce, they can pass, when they reproduce, they will pass their genes to their next generations. So they will get the resistance gene. And now most populations of the protozoa are resistant against the disease. So what's the solution? The problem is the malaria medicines no longer work because this malaria or this protozoa is getting stronger, so they don't work. So how can scientists use another idea to fight this disease? The best cure is an expensive chemical called artemisinin. These artemisins come from a plant called Artemisia, okay? So they need to extract this chemical from the plant, but it's so expensive because it's hard to extract. And they need more. If they want to grow more of this Artemisia plant, it will take too much to grow, to extract, to get in a specific stage to get this chemical from it. So they need another solution, okay? And also to make the artemisinin against the disease also, so the disease, or sorry, the protozoa cannot develop another resistance against this artemisinin. They need to mix it with another chemical to make this medicine, okay? So in this case, microbes rarely develop resistance to more than one chemical at once. So the protozoa can be resistant to one chemical, but not a mixture of chemicals. So that's why sometimes they need to mix more than one chemical in one substance to make it hard or to make it difficult for the microbes to develop a resistance against them. The problem is that, as I said, it costs a lot because it's difficult to extract. So what's the solution? Scientists used genetic engineering and selective breeding to produce more, or to develop the genes, or to get more of these genes. Okay, what about the selective breeding? The amount of artemisinin in the plant leaves vary. So when scientists studied or checked thousands of Artemisia plants, they found the amount of artemisinin in their leaves varied, which is different. Some of the plants are high, some of the plants have low, and some of the plants have medium percentage. So, but here, few plants gave a much higher yield than the rest. So this is an advantage, right? that they have high percentage or they have high yield than others. So how can they take this advantage and use it for them? Guys, can you?
They can breed they can choose the ones that produce more and more artisans in it,
Okay.
and like breed those two together. So the next generation will produce a lot of this.
Okay, thank you so much. So they will breed them, the best plants together, so they will produce new varieties with even higher yields. So if they get these genes, the best genes together, they will produce higher percentage of this artemisinin. In this case, they will get more of this chemical. Okay. This is about selective breeding. They can use another way. Why selective breeding is not that much successful sometimes? Because it can take time to grow the plant, to breed them together, to provide the suitable conditions for them or the environment for them, to wait until they grow, also to extract, because it's, as I said, it's difficult to extract this chemical from the leaves. So it will take more time to get this chemical. So they can use another way, which is the genetic engineering. We said genetic engineering, depending on moving the genes from one cell to another different cell, which is another organism's cell. Usually they are using the yeast. Why they're using the yeast cells to produce artemisinin? Mohammed, can you tell me?
What?
Can you tell me why most of the time they're using the yeast cells to get this artemisinin? What's the special about the yeast cells?
Yeast cells kill bacteria?
No, yeast.
Yeast.
Do you know what is yeast?
Yes.
Is it
okay.
Is it spot?
When you add the yeast to warm water with sugar, what can you see?
Reaction.
What? No, we don't call it reaction.
It will multiply very quickly.
Yeah. It can multiply quickly, fast, reproduce fast. So that's why they are using this yeast. So scientists, they found the genes that plants use to make this chemical from the plants. Okay, they found the genes, so they add them in the yeast cells. Since the yeast cells easy to reproduce and multiply, they made them produce artemisinin. So when they reproduce, they will use this gene to produce artemisinin. So here, we have the living things, all the living things can read each other's genes. Why? Because when they add these genes to the cell can read it, or the nucleus can read this gene and produce more of it. If this gene is safe, doesn't kill the creature or this organism, it will just pass it to or multiply it and reproduce more of it. So here, they can copy these yeast, they can copy their genes quickly and make lots of proteins these genes code for. and reproduce quickly. In this case, they can reproduce for artemisinin. Okay. Since this takes a lot, takes a lot of expensive to make genes work probably in another living things, but other medicines have been made like this for many years. So in selective breeding or in extracting this chemical, which is the artemisinin from the Artemisia plants, is expensive and take more time, more money. Most of the medicines, they were made by the same way, which is the genetic engineering, because it's easy to make this organism reproduce quickly. All over the world, the microorganism, the yeast cells, sorry, they are reproduced and grow in a huge tanks, like the bacteria. When they want to grow bacteria, they're using very huge tanks to grow them, and then they can extract the chemicals or the chemicals that they want from them. The same thing with the yeast. They're using a huge tanks now all over the world to grow and make them reproduce. Artemisia plants take more than 1 year to grow. This is the difficulty in growing this plant and using selective breeding with this Artemisia plant, because it takes more than 1 year to grow. But here, the yeast is easy to reproduce, so it will be easy for them to extract the artemisinin from them. And also,
is
it's
yeast
cheaper.
a single-celled organism?
Sorry?
Is yeast a single-celled organism?
Yeah, they are still they have only one cell. Okay, so as I said, it's also cheaper to extract it from the yeast other than the leaf itself. Okay, now we will talk about stamping out malaria. Like, how can we work on this medicine to stop the malaria or to fight the malaria? The governments and charities all over the world work together to tackle the disease, like to attack the disease or to stop trying to deal with